All right, this is a very great topic. The main motive is to take away important points so that you know what the basic disease process starts with. So, we are delving into oxidative stress. Here, each molecule could turn out to be a great mess in our body. Even oxygen can be formed into superoxides, which would then be converted into hydrogen peroxide. Now, superoxide dismutase is a very great enzyme that kind of regulates the oxidative stress that is happening. We need these metabolites so that they are inactive and eventually there is no oxidative stress. Okay, there is a half life. This is something very important. It won't stay in our body. However, the end products are very, very important. Let us say if the superoxide radicals are very high, or let's say nitrous oxide is very high, then there can be impairment in the functioning of how our body actually has to function. This can create highly reactive substances leading to nitration, or through Fenton's reaction there can be lipid peroxidation or protein peroxidation. All in all, the molecules can turn into a abnormal shape, resulting in a damage to the DNA. And finally, there can be cell death. Overall, our main motive is getting this oxidants to a lower level. If it is heightened in our body, there can be response proteins, there can be a cellular stress that is happening. And to combat these, we have antioxidants in the market. Okay, so antioxidants are something that I will be discussing in the end of the video, probably, but you just look at how much of chemical reactions our body is going through. Previously, in our 12th grades, we learned ETC is one of the main cycles where electrons are derived, oxygen is derived. It's like the main source of superoxide radicals. Now, we need to know that there are mechanisms that come back to this to regulate the oxidation and nitration. Furthermore, it is important to always remember lots and lots of hydrogen peroxide will form hydroxyl radicals. This in turn can lead to modifications, leading to cell death in the end of the day. Similarly, we have glutathione, that is oxidized form and the reduced forms. So, in the oxidized forms, there can be lots and lots of oxidative stress that is happening. So, there are drugs that typically target this particular enzymes. Okay, I'm not going to go into details about what happens and so forth. Let's move directly to the conclusion of what the authors are suggesting about. Yes. So, in summary, we need lots and lots of molecules for us to build it into proper proteins, because proteins can make its way to the brain and it can be generated as a functional component. What happens in Alzheimer's disease? There is lobes, like hippocampus, parietal lobes are shrunken because the proteins that are being produced are way different compared to the aged individuals. Okay. So, technically, we cannot make out these important ideas until and unless we go through a biopsy result. Uh, in the body, any organ can be biopsied, any invasive procedure can take lot of risk along with it. Similarly, if we want to get a tissue from our brain, it's highly unlikely process because going through the skull and then placing the electrodes and taking a pinch of that brain is deaf brain or a piece is not an ideal way, right? We can take a biopsy of the brain to know what is going on. So obviously after death there were a lot of autopsies being done on particular person with prior consent. So postmortem intervals were being done like every 4 hours they checked. In this paper, the author also suggests that there are meaningful conclusions to establish that oxidative damage to the brain is not as advocate. Meaning, we have a higher bar set for patients with Alzheimer's compared to the mild cognitive dementia patients. Um, the amount of proteins are different. The density is different. The structure is different. If you ask me, are the proteins made in aged individuals? Yes. If you ask me, are the proteins of same structure in the aged individuals compared to the AD? Maybe yes or maybe no. If you ask me, what is the protein nature in AD patients compared to the aged individuals, pretty much the same. Okay, so for proper distinctions, the authors or let's say the researchers will need a demarcation to say yes, this is abnormal. This is a very pertinent study where proteins were being studied in detail. And as you can analyze from what I have spoken so far, proteins are some of the great essential components for the oxidative mechanisms in the process of getting out the metabolites. Sometimes these proteins are modified towards a product that is reactive, meaning it's way too reactive. And that is what leads to hyperphosphorylated tau proteins. Okay. And slowly, these tau proteins will find its way to the brain across the lobes. And this is what leads to progression of the disease, which can be made out through the imaging scans. Now, the next point I like to highlight is the appearance of common targets. Further implies that there are important roles that are set by the authors in this paper. Um, it can be a mitochondrial dysfunction. There can be short synapses where the communication could have been hampered, or there can be neuroinflammation, meaning the brain is also inflamed. Like, for example, if there is a body inflammation, the vagus nerve carries the signals all the way across the blood brain barrier. Okay, so those studies are also being done recently. And it's clearly a great way to correlate the person who has had dementia with that of the AD patients. The key here is the oxidative damage that has been ongoing in a patient with AD compared to that of the aged individual. Conceiving this idea and taking forth this idea forwards into a proper direction, they came across great strategies to identify what is causing this reactive species, reactive molecules, or whatever you call. Okay, so I'm going to discuss this further in the next video. Stick on with me and we will go through it together.